Good afternoon, everybody. Yeah, it's not on. Sorry. No, it's on. No. Yeah, good afternoon. I'm Sebastian from Aarhus University in Denmark. So I'm really happy to share with you this story about how in Denmark we have been using soil microbial data to understand soil health indicators from a mapping perspective. Maybe you already know how important the soil microorganisms in terrestrial ecosystem. This is a very simplistic view and a very simplistic figure about this importance, but just to highlight some of the most important functions related to carbon and nitrogen cycling and also energy fluxes in terrestrial ecosystem. So we know that they are very important, but we don't really know how to use this information and to integrate this into soil health assessment frameworks. Mm, from a some regulation and some regulation perspectives, now the soil monitoring law is including soil biodiversity between the list of soil health indicators or descriptors. You can see last but not least is the soil biodiversity. And according to literature, we can express this biodiversity in different measures or representations, specifically for microbes in soil. For example, microbial biomass, enzymatic activity, community composition, the presence and also the relative abundance of functional groups, or there's also the option to represent as nitrification or other processes and the corresponding rates, and also the relative abundance of microbes that are beneficial for plants. Some interesting studies from the mapping perspective. So we can see some very few examples on people previously tried to map different representation of soil microbial diversity across different study areas. But definitely the most interesting part now is that DNA sequencing has greatly facilitated the studies exploring the soil biota and the composition and also the dynamic. Thanks to these new methodologies and some advances in computation and all of these things, some colleagues in other countries, in other continents even, and has been um, allowed to map this representation of microbial diversity. The point is that we know that we have some tools and we have capacities to map these um, indicators out. But the thing is that that based on these exercises, we need to unveil the main drivers of microbial diversity in soil, but we also need to understand how to use this to assess soil health. And that's the point of this presentation and the point of this story. So let's go with some data and methods. We have around 7,000 samples collected in across all Denmark in different agricultural and natural habitat types. You can see here in this figure the distribution of our points. And with the help of some of our partners in Denmark, they have been doing a lot of metagenomics and all of these bioinformatics things. So they have used shallow shotgun metagenomics to generate information for us in Aarhus University to be able to map these indicators and study a bit more in deep about it. So that's just an example. That's what we got from them. It's a very, very huge table when we have all of our points, coordinates, and we also have a relative abundance of different taxa. In this case, it's relative abundance of genera of prokaryotes. So now we need to figure out how to translate this table into meaningful maps or meaningful spatially explicit information to understand the soil health. So the first question was, what can we map with this huge, huge data set? So we can go from the very simple indices to a very, very complex representations. So we started with one of the most common from the ecological perspective, that is this Shannon diversity. Is very important and is very commonly used in ecology. So this is the thing that we first did with our table to calculate these indices and also others, but that's the most common. But then the point is that we can get, and according to the presentation this morning, the keynote speaker mentioned that it's very difficult to figure out what is the best index to represent the soil diversity. And also, it's a very interesting point that we need to see some what is the best way to represent not only the amount of organisms that they are in the soil, but also the functions that they are performing. So we tried also to couple our database with another database called FAPROTAX to relate the prokaryotic taxa to ecosystem functions. So it's a bit of uncertain, this relationship between these two databases, because FAPROTAX is mainly based on literature, but it's also a kind of use that we try to do with the data. And this is just an example of the relative abundance of methanotrophs. So we have also in Denmark some environmental layers. You can see some examples here. We have elevation, slope, soil textural class in a very coarse representation, and the presence of organic soils. Along with these maps, we can also include, or we could also include some other soil properties, bulk density, texture, pH, organic carbon, some topography derivatives, organisms to represented as satellite bands and land use maps. And in a very usual framework of digital soil mapping, we try to use machine learning models along with these layers to produce prediction maps, uncertainty maps, but more importantly, to try to understand what we are mapping. So that's a very important part, is understand the predictions, interpret them, and then it's like a baseline to figure out how to use the information. So let's go with some findings. So it's an ongoing job. So some partial findings. Let's see Shannon diversity maps. So I will show you the prediction map and also the variable importance like this. Shannon index, we have different values. It's an interesting pattern for us, for Denmark, for this alpha diversity. So we have low values in Western Denmark, but also in the central part and also in southeastern Denmark. And these values, these spatial patterns are related to the variables that you can see in the right. Basically neutral to alkaline pH zones with these pH and vegetated areas, mainly croplands, are related to higher values of this Shannon index. In contrast, we found that enclosed areas with sandy soils, especially in Western Denmark, but also clay soils, especially in southeastern, in the southeastern part, are related to low values. But we also found that heathlands, the presence of heathlands is driving some low values in Western Jutland mainly. The other thing is the relative abundance of methanotrophs, also prediction and variable importance. We can also see an interesting patterns here in the map on the left, and some of the important variables driving or influencing the spatial patterns of this relative abundance. So we could see that, for example, land use has a very strong impact along with soil properties. And that's what we have learned so far from these maps and other examples, but specifically from this, that soil and properties and land use are playing an important role in this. Mm, but then now what? So what's next? So our perspective is that we can use this information to couple with some exercises that we have done before. Basically, what if we analyze the distribution of these indicators within homogeneous zones? Or in other words, what if we benchmark these soil health indicators, assuming that these can be called as that within soil monitoring units? So previous works also we have done in these soil monitoring units to generate them for Denmark, some exercises that we have done, mixing or combining texture and landforms and land uses. So, and based on points, but also prediction maps. Let's see how it looks like for prokaryotic diversity in soils. So we have, for example, this distribution for the whole of Denmark, and we have different distributions of this variable across different homogeneous zones according to previous exercises. But let's see, for example, the plot in the right. So values considered, for example, as typical in sandy soils in flat areas, uh, under grass in rotation, for example, might be low in a different context. So it's important to see this, how this can work with the prokaryotic diversity, but other indicators as well. And since this is an ongoing work, so it's an ongoing wrap up. So very important to interpret machine learning models to enhance our understanding. So far, we have seen that soil properties and land use are very important drivers for it. But then now we need to think a bit of what is good or what is bad in terms of microbial indicators for soil health. Yeah, and I think my time is over. So thanks. Yeah.